from a bacteria to a blue whale, it's the same 4 letters, A, T, G, C, the DNA. All living things. It's housed in their nucleus or the nuclear area in a prokaryote. Just pay attention back there. All right, so what we are doing is, in science, and in particular biology and chemistry, we have means of separating things out. And that's what we're doing. This is a separation technique. We're going to pull the DNA out of that strawberry by following some simple physical steps and some chemical steps. Does that make sense? All right, so I'm going to come back over this. We're low on time, but these are what the chemicals do. The soap, the salt, they're going to react. See, the soap's going to react with the cell membranes. If I go back a sec, there are enzymes that help break down pectinase and cellulase. Those are enzymes that break down the cell wall. So a cell wall, they're plants. We don't have a cell wall. So it's an added step. When they get ripe, they start breaking it down. All right? We're going to try to extract the DNA, and here's all the steps. Okay? Freeze this. All right? And I'm going to go to the back, and I want you to unfreeze it. So come on back here. All right, so first, we got our little baggie here. So I'm going to come back here and point out a couple things. All right, so here's our setup. After we have our mixture that's going to be in here, I'll do that last. We're going to shimmy it down. We're going to pour it into this. We're going to filter it. Okay? So let's just kind of watch there. Start at the beginning. If you look at the steps over there, why don't you come over here? Okay? So first you got your strawberry. The strawberries are where you're about to back up into. Whatever this pile is, you're going to pick up your strawberry, drop it in. And you're going to start the first part, maceration, mashing it up. Okay, so you're going to mash it up. Mashing it up is this, right? Not this. Nothing like that. See, I popped it. That's what I'm trying to avoid doing. All right, and if you end up having a little hole there, then what you have to do is seal this end off, and you still can be mixing it up. We did have some people like this. We do not want that. Although it's a cool sound, the person wearing a suit today in first period didn't really appreciate getting strawberry all over them. Okay, so when you're mashing it up, so because my bag is ripped, assume you got the reverse, so this is it. All right, you got it in there, you're mashing it, then you start mixing it around. If you look at the next step, you're going to add a squirt of liquid soap. Okay, I have liquid soap over there, if you look over there, for you two groups. For these guys, you're going to use this, and I had a bottle over here somewhere. I'll get it for you. Oh, grab that. That's for the. So I got soaps for each of the groups in each row. A squirt of soap gets mixed in here. You start massaging, mixing it in. And now this is where the point where it's mashed up. You got little bits up there, little smattered bits of destructive strawberry that's been macerated. I know that sounds very dramatic, but that's what it's going for. All right, so then you pull it, shimmy it back down to you get, like you say, your corner. All right, then a pinch of salt. Thanks, Alice. And we have salt. You just take this whole cup, dump it in there. Not this one. This was the source one. So move that out of the way. Okay. So then you're going to keep coming down, mixing it in, mixing it in. All right. So it says now add some water. Remember the picture I showed you earlier? So you add some water. So let's say, look at my finger. That's like the level. It's probably no more than that. Go ahead and unfreeze. Okay, so now look, we're at step 6. This is cheesecloth, all right? That's the filter. Come on back over here. You got me a second test tube. Okay, so here's the deal. You're going to dump all the contents in here, and now what's going to happen is, and now you're looking top to bottom, but I'm just doing this for the camera. What happens a lot is, you got all this stuff here. Some students first period did this. They kind of moved it around, and it helped it go down through a little more quickly. So what's going to happen is if you add too much water, once you get to here, every group should have a second overflow test tube. So what you'll do is just quickly take this and move it over to there, let it keep filtering. Once you're about to here, then you're ready for the alcohol, rubbing alcohol. Okay, over here, these are empty beakers, but by the time you guys start filtering, I'm going into the room, getting the alcohol out of the freezer, and I'll pour it in, and you're going to take this, we should be wearing goggles, and you're going to dump your rubbing alcohol into out here. So let's say your filtrate, that's what is your mashed up strawberry, the ruptured cell walls, the ruptured cell membranes, and then the chemicals that are pulling pulling all that stuff out of the cells into a solution. Then you filtered it through. All the bits you leave there. This should be like a liquid. All right, should look like strawberry juice, not strawberry pink lemonade. And so, say when it's about here, you're going to take and add rubbing alcohol up to about the white spot. All right, you may have some left over. Just bring it back, and then we're going to extract it. What are we going to do? We're going to extract it. See, this DNA does what we call precipitate. It precipitates out. It comes out of solution, and this is a separation technique. It's going to float to the top. And all you're going to do, and this part's optional, but when you see it flowing up, basically you're going to dip the little things, twirl it around, and pull it out. And it should look like snot on a stick. And that is the goo, that is you. All right, from a bacteria to a blue whale, the same 4 letters all exist. A, T, G, C, the DNA. From a bacteria to a blue whale. The difference is the arrangement, we call that the sequence, and the number. All right, and that's what we're going to be exploring over the next couple of weeks. All right, go ahead and cut.